has just a really amazing track record and history of publishing really great science and doing great work in melanoma. So he did his PhD at the Moffitt Cancer Center in the lab with Kieran Smalley, really focusing on MAP kinase targeted therapies and also mechanisms of resistance. He did his postdoctoral fellowship at UPenn, where he really focused on autophagy and lysosomal mechanisms in cancers, including melanoma. He then transitioned and was a scientist at the White Star Institute, where he worked very closely with Maynard Herlin on not only just developing and expanding PDX models, but also continuing his work on therapy resistance in melanoma. So now for his lab at Hopkins, he's kind of taken what he's done in melanoma so far with therapy resistance and sort of autophagy, catabolism, and is now really focusing a lot on acral melanoma. And as he'll tell you about today, acral melanoma is rare type of non-sun related melanoma, and it also affects persons of all ethnicities and background. And so just before I hand it over to him, I just really want to say I'm also really honored and really impressed by his work in the DEI field. And so really promoting diversity and equity and inclusion in the melanoma science field, as well as for these rare types of melanoma. He's started a specific workshop at the annual Society for Melanoma Congress on DEI, and he's got numerous publications on DEI. So he's also received a number of awards from SMR, the Christopher Marshall Award, as well as last year, the Melanoma Research Alliance Young Investigator Award. He's got 2 first author publications in cancer discovery, 2 co-author publications in cancer discovery, numerous other co-author papers in Nature Genetics, Nature Communications. So I'm really excited to have him here today to talk about his work in acral melanoma and just broadening your knowledge and insight into the rare melanoma world in general. So thanks for being here today.
Thank you, Casey, for that really nice welcome. And thank you for everybody I've met with today. I've loved hearing everybody's exciting work, and it's a pleasure to share our lab's work. I'm going to be sharing something we just recently published and something that we're trying to put together for publication. So any feedback, insight, we would really appreciate. So our laboratory focuses on melanoma, and so this arises following the malignant transformation of melanocytes. And so melanoma is a major burden to public health. It remains one of the few advanced cancers whose incidence continues to rise. In the year 2023, it's estimated that about 100,000 new cases will be diagnosed, and it cost the U.S. healthcare system more than $5.7 billion. And so melanoma arises from melanocytes, and an interesting thing is that melanocytes could be found throughout your body. And depending on where anatomically that melanocyte exists, it dictates the type of melanoma you can get. And so here is just a schematic of the different subtypes, different sites. The most common types of cutaneous melanoma, at least in European Caucasian populations, are lentigo maligna, nodular, and most commonly superficial spreading. And so this is the subtype. If you've heard of BRAF/MEK inhibitor therapy, immune checkpoint blockade success, it's been predominantly in the superficial spreading melanoma patients. What is the rarest form in Caucasian European populations, what actually represents the most common melanoma subtype in communities of color, is acral lentiginous melanoma. And so acral melanomas arise on distinct sites where the sun doesn't really shine, and that includes the palms of your hand, the bottom of your feet, and fingernail beds. If you have a melanocyte in the eye that becomes transformed, you can have a uveal melanoma. Also, different mucosal sites, as Casey and many of you here study. You could also get a mucosal melanoma. These represent also very difficult to treat situations, but our laboratory primarily focuses on acral lentiginous. And it actually represents a much more lethal subtype compared to other subtypes of cutaneous melanoma. And so in addition to being more lethal in general, so here, before I get into the details of our work, I just want to introduce the people that actually have created all this work, our A team, and the investigators in red are the projects, the people that have been pushing the projects I'll be sharing today. And so this is our laboratory, and then we have a lot of collaborators across Johns Hopkins that also help us make this work as meaningful as we can. So why do we study acral melanoma? Patients with metastatic acral melanoma suffer 5-year overall survival rates of less than 16%. So this is a bit worse than other subtypes of cutaneous. Preclinical research of acral melanoma is nearly non-existent. The drivers of acral metastasis remain to be identified. As such, the drivers of acral melanoma therapy resistance also remain to be defined, and racial disparities persist for these patients with acral and have actually worsened for Hispanic and non-Hispanic Blacks compared to non-Hispanic whites from the time period of before 2000 to the after 2010 period, when a lot of these breakthrough therapies have been FDA approved. And so when we look at the racial disparities of acral melanoma, in general, it's more lethal than other subtypes of cutaneous. But if you stratify the acral melanoma patient population by race ethnicity, you see that patients that are Black, Hispanic, white, or Asian Pacific Islander suffer worse outcomes compared to patients that are non-Hispanic white. And if you normalize the tumor thickness and the stage of presentation, these trends typically also hold, suggesting that there might be biological drivers for this phenomena. So our laboratory, in addition to all the science, we're also interested in trying to, do what we can to prevent these racial disparities from occurring. One of the things might be differences in communication of the risk into different populations of color. And so a medical fellow in our laboratory, Visi Elad, actually investigated what's out there in the medical text regarding melanoma and acral melanoma. In general, when you write something for the general population, the recommended reading level that you write these information, details is supposed to be at around a 6th grade level. But if you look at Spanish texts for medical warnings of melanoma, as well as English, you see that typically they're written at a much higher level than what would be ideal for the general population. And so Visi in our lab are trying to come up with infographic, little cartoons that can better give that warning to the general public so that people of color can realize melanoma comes in different types, and there actually are risk areas to check out. And so this is just a little story that we've done. So today's talk, I'm going to be breaking it apart into first our efforts to try to understand the metastatic drivers of acral melanoma. Part of the reason why the acral melanoma metastasis driver landscape is so poorly understood is at the moment, genetically engineered mouse models of metastatic acral melanoma do not exist, and an orthotopic mouse model of metastatic acral melanoma does not currently exist. And so this is an area our laboratory for the last 2.5 years is focused on to try to create a first-in-class orthotopic mouse model so that we can begin trying to understand how these tumor cells end up in metastatic sites when they begin in the foot. So to begin these studies, what we did was we first took advantage of pre-existing GEM models of mouse melanoma. So these are the Yum 4.1 and Yum 1.7 created by Marcus Bosenberg and Martin McMahon. And so Yum 4.1 was the first one we began with because its genotype is similar to an acral melanoma patient. Its BRAF wild type has a CDK4 pathway alteration. And so potential significant differences. So when looking at the ability of those primary tumors to grow, what we observed is typically the tumors in the foot grow much slower than those tumors in the flank. This might be a virtue, there's less space in that foot environment, but we're exploring what could be driving that maybe biologically. But the really exciting thing is when we looked at for metastatic differences, we found that those Yum 4.1 cells, we did not see any evidence of metastasis in the lung, liver, brain. When we looked at those Yum cells implanted in the foot, we saw that 4 out of 5 mice displayed significantly, liver metastatic phenotype. So of course, we wanted to see, could we recapitulate these metastatic findings using human acral melanoma cell lines? And so we again used the same approach. We took 3 acral melanoma cell lines with different genotypes representing as much of the patient population as we could. We functionalized them with, again, the M-Cherry luciferase tag. And this time we used the immunodeficient model. And so we, again, observed that those acral melanoma cells in the foot grew typically slower. We have these experiments ongoing. It takes, for whatever reason, more than 3 months for something to grow. But the same trend of a slower primary tumor growth rate. And again, we found, in this case, elevated metastasis when we checked at the lungs and the brain of these plantar foot injected acromelanoma cells compared to the flank, where the only evidence was 1 out of 5 mice a lung. So clearly there appears to be some kind of priming by that plantar skin microenvironment of an elevated metastatic state, and our lab is very interested in trying to understand why. And so to begin this characterization and verification, we first performed primary bulk RNA-seq of those primary tumors implanted in the plantar skin of the foot versus non-plantar skin. Something we observed was that there was a significant elevation of SOX10 in those tumor cells grown in the plantar skin versus the foot. And something that's interesting is there's been some evidence that in acromelanoma patients, SOX10 high acromelanoma cells are immune evasive and less likely to respond to therapy. We also saw an enrichment of HOX13 in those foot pad injected tumors. This is interesting because there was a recent publication in 2022 by the Richard White lab where they found that acromelanomas that overexpress crackle, they typically end up in the fins and the tail of zebrafish. And when they checked other gene signatures that were associated with a crackle high, they found that HOX13, which is associated with limb development during development, was also enriched. These tumor cells have never been grown in the foot versus the flank. So this data suggests that perhaps tumor cells might have an ability to sense their local microenvironment and know anatomically where they are in the body. We also checked then some gene set enrichment analysis, and we found that there was an increase in invasive signatures, cell motility signatures. We also found, as one of the most significant enriched gene programs, a collagen degradation signature, which we began to be excited about because that plantar skin of your foot is defined by increased abundance of collagen, increased thickness of collagen. And so it seems to us perhaps the acromelanoma cells were adapting to this unique collagen environment for their survival. And so in addition to that collagen degradation signature, we saw evidence of increased cathepsin K, which is a lysosomal enzyme capable of being secreted by the lysosome into the extracellular space and cleaving collagen, which would then allow tumor cells by macropinocytosis to internalize and recycle that collagen. And there's evidence in other tumor types of cathepsin K fueling metastasis. And so, of course, we wanted to check at the protein level, and we see that there's increased protein expression by IHC of cathepsin K in those plantar-embedded tumors versus non-plantar. Cathepsin K can be active and inactive depending on the cleavage, and so we check by Western blot and can confirm that those tumors in the plantar skin are indeed expressing high levels of active cathepsin K. And so we have a recent grant looking at testing the hypothesis that cathepsin K is what's driving that elevated liver metastasis phenotype, and if we're going to test whether or not inhibiting cathepsin K can prevent that from occurring. So that's something that's in the works right now. We're also interested in trying to understand how that plantar microenvironment might be epigenetically priming the acromelanoma cells to an elevated metastatic state. So to that end, we saw some gene set enrichment analysis showing increased, significantly different changes in histone modification, differences in chromatin remodeling, as well as differences in histone deacetylation. So in collaboration with Anita Tosca, who's a breast cancer epigenetic expert at Hopkins, we're currently performing ATAC-seq on these tumors from the foot versus flank to try to understand how that microenvironment might be changing the epigenetic landscape toward metastasis. So we have some theories of how the plantar microenvironment is increasing metastasis. As I mentioned, the skin of the foot versus the skin of non-plantar skin has significantly elevated matrix stiffness. There's significantly greater collagen density. And so we're testing the hypothesis that perhaps the mechanotransduction pathway with integrins able to detect differences in stiffness and collagen density may be hyperactivated and might serve as a vulnerability of these acromelanoma cells. We're also testing whether or not that cathepsin K lysosomal catabolism might be serving a role toward that elevated metastasis. And so some techniques we've been doing to just verify what is known in human skin is occurring in our mouse skin. We performed CODA, a brand new technique invented by Ashley Kimmel, an investigator at Hopkins, as a PhD student. Now she's an investigator at Hopkins in the laboratory of Denny Wirtz. And so how CODA works is it allows you, there's supposed to be a video. CODA, compared to how standard IHC characterization occurs, where you take a single slice or two of a tissue, with CODA, you take over 100 slices throughout the entire block of tissue, and then every third slice you H&E stain, and you're able to, using algorithms, create a 3D map of that skin to appreciate all the heterogeneity in different regions of that tissue. And so using that technology, we've begun to profile non-tumor bearing plantar skin of the foot versus the flank to really see the differences in the architecture of the microenvironment. So shown here are some of the different characteristics you can get out of this technique using these computer algorithms. And we can confirm that the skin of the plantar skin of mice versus non-plantar skin, there is a significant increase in that collagen volume. We've also confirmed whether or not there might be differences in the stiffness in our mouse system that mirror the human system. And so in collaboration with Luo Gu, who's a materials engineer expert at Johns Hopkins, our PhD student Bailey collaborated with his PhD student Supeng to isolate the skin of the foot versus the skin on the flank. And using this cool tool, the Puma microindenter, Bailey was able to confirm that indeed the skin at the bottom of the foot is significantly stiffer versus non-plantar skin. And so to test, there's the video. So you cut all the way through it and then you make that nice 3D map. Bailey in our laboratory, as part of her PhD work, has begun then trying to investigate whether those differences in the matrix mechanical properties might play a role in melanoma biology. So what Bailey did here is she created a series of 3D spheroids of acral melanoma cells, and she implanted them into collagen plugs of increasing density. And she can confirm that it appears that as you increase the collagen density toward a plantar skin microenvironment, those acral melanoma cells become much more invasive. When looking at stiffness, we collaborated with the laboratory of Costas at Hopkins and his PhD student, Alice. And so here, Costas has created these microfluidic devices that have channels of a specific size, and you can determine the rate at which your, in this case, acral melanoma cells can squeeze through those tiny pores. And so what's really nice about this approach is that Costas can functionalize the chip to have different stiffnesses. And so as you can see here, as you increase the stiffness of that matrix, the acral melanoma cells also exhibit increased motility. So this is what we've had so far in terms of our approach to understanding the metastatic drivers of acral. We have a phenotype that those tumor cells implanted in the plantar skin appear to be primed toward a greater metastatic capacity. The plantar skin microenvironment, specifically collagen and stiffness, appears to induce cell motility, invasion, lysosomal catabolic gene signature. We're at the moment trying to understand what vulnerabilities we could attack to prevent those phenomena from happening. And so for the next part of our talk is our efforts to try to understand the therapy resistance landscape of acral melanoma. And so when we look at the superficial spreading melanoma, a ton has been done. These are just a handful, really, a few of the resistance mechanisms that have been published as cutaneous melanoma cells escape various forms of targeted immunotherapy. These include receptor tyrosine kinase upregulation, BRAF, CRAF, isoform switching, metabolic reprogramming, post-translational modifications, hyperactivation of parallel pathways. All to say, when we look at acral melanoma, it's a completely wide open landscape. Nothing has been published in the way of a concrete resistance mechanism with preclinical work. And so we're excited that 2 weeks ago or so, our lab's first paper was published to begin filling up these gaps in terms of how these cells might uniquely escape therapy. And so some reasons why acral melanomas might be more lethal compared to cutaneous is they have a lower frequency of BRAF mutations. Compared to superficial spreading that have about 50%, there's only about 15 to 20% in acral. This is important because there's FDA-approved treatment of a BRAF plus MEK inhibitor that is really effective, and unfortunately, the majority of acral melanoma patients are ineligible. So in addition to that, a story we published with a clinician at Hopkins, Crystal Lagu, we also found that those few BRAF mutant acral melanoma patients that did receive BRAF or BRAF plus MEK inhibitor therapy, they also did not respond as well as the cutaneous BRAF mutant counterpart for reasons that are not clear. And so a PhD student in our laboratory, Marie Portualo, is investigating specifically how BRAF mutant acral melanoma cells might respond differently to BRAF mutant superficial spreading melanoma cells. In addition, acral melanoma, because it arises on areas that the sun doesn't shine as much, there's a lower mutational burden. And so this is correlated with a lower response rate to immune checkpoint blockade. Because there's lower mutational burden, there's a hypothesis that that decreased neoantigen repertoire is maybe why the T cells can't engage as much. But we're investigating also whether there may be unique properties of that skin microenvironment of the foot that might be also promoting this more immune evasive and poor response to immune checkpoint blockade. And so a master's student in our laboratory, Samaya, is looking into this. But overall, it's clear that effective therapy strategies are needed that are tailored specifically for acral melanoma patients, because what's been discovered and used in superficial spreading melanoma is not working to the same extent. And so our laboratory, to begin figuring out what would be a good place to begin building a better strategy, we first focused on CDK4 inhibitors, because there's been evidence that 60 to 80% of acral melanoma patients have CDK4 pathway alterations. So this is a great number, have amplification at the gene level of cyclin D, CDK4, CDK6, copy loss of CDKN2A. And overall, this prepares the cell to be in a very highly active cell cycle state. There's also evidence that these alterations in the CDK4 pathway might be functional, because acral melanoma patients with evidence of CDK4 copy number gain have worse survival compared to CDK4 normal patients. And so this has been part of the rationale to investigate using CDK4/6 inhibitors, such as palbociclib. There was also some evidence from this group in 2017 that PDX models of acral melanomas with copy number alterations were sensitive to palbociclib, and those PDX models with wild type were not. And so based off of that one study, only acral melanoma patients with evidence of CDK4 pathway alteration were eligible for this first-in-class CDK4 inhibitor trial. All to say, the median progression-free survival of that trial was 2.2 months, which demonstrates that rapid acquisition of resistance is likely occurring. In this trial, it did not consider RB expression. This is commonplace in breast cancer. Use of CDK4/6 inhibitors, because of course, what a CDK4/6 inhibitor does is activate RB to stop the cell cycle. So that could play a role in why this drug did not work as well. In some centers of this approach called a basket trial. And so what a basket trial does is it tries to better personalize therapy. At the moment, if you have a cutaneous melanoma and you have a BRAF mutation, you're given then BRAF plus MEK inhibition. But it's clear that not all patients are responding the same. And so in an attempt to better personalize, this approach has been devised where they perform targeted seek on patients to see, in addition to a BRAF mutation, what other mutations might you have. And if you have a mutation that there's a drug against, you would receive a BRAF MEK plus a combination of that drug inhibitor. So for example, if you had a BRAF mutation and an AKT mutation, you would get a BRAF MEK inhibitor plus something that hits the AKT-mTOR pathway. And so all to say, this seems largely like a great idea to better personalized therapy, increases your chances of getting a response. The evidence from the clinic is not really showing that it's making a big difference. Have greater phospho-AKT activity at the protein level. Same thing, if you have a greater copy number variation, you would predict that the mRNA is elevated, and the mRNA would be predictive of pathway activity. And I have little question marks because it's not been really robustly fleshed out that these assumptions at the genetic level are actually being found at the protein level. And of course, the final important assumption is that all of this information would predict sensitivity to the inhibitor against that pathway. And so using our system, I'm going to show you some data here. We took advantage of targeted sequencing of the CDK4 pathway at the gene copy number. We have the RNA information, we have the protein information, and we have the sensitivity to really put to the test, is this a good idea of how we should stratify patients for treatment? So to begin, we took a panel of acral melanoma cell lines. So here are a panel of acral. These are non-acral superficial spreading melanomas. And, mirroring what we see in the clinic, there's a high degree of CDK4, 6, and cyclin D amplification in these acral melanoma models. We then performed protein expression analysis by Western blot of baseline cells. And then we wanted to see some correlative studies to see how well is a copy number amplification predicting increase in protein expression. And so when we look at CDK4 protein expression on the Y versus the copy number status on the X, we did not see a significant correlation. We also did not see that CDK4 copy number was predictive of protein expression. And also with cyclin D was not predictive of protein expression. And of course, you can see there's not many samples in our system, but we took advantage of the TCGA, where there's about 100 cutaneous melanoma patients where the copy number status and the protein expression by reverse phase protein array is known. And when looking at cyclin D, we also did not see a correlation with protein and copy number status. All to say this, assumption that copy number is predictive of protein expression and therefore potentially sensitivity, at least for acral melanoma patients, it might not be the case. And so we then wanted to understand how are these acral melanoma cells escaping therapy. And so we used palbociclib, a approved CDK4/6 inhibitor. We analyzed treatment by reverse phase protein array and see all the cell cycle machinery downstream of CDK4/6 was being inhibited sufficiently. We also confirmed by Western blot that in a dose-dependent manner, all the cell cycle machinery was being inhibited. However, when we looked at the ability to inhibit melanoma cell proliferation or growth in 3-day assays, we found that palbociclib, as well as 2 other clinical grade CDK4/6 inhibitors, ribociclib and abemaciclib, there was still a significant number of these acral melanoma cells growing beyond the physiologically relevant dose of 1 micromolar. We also performed 2-week to 3-week colony formation assays, where here in orange is the cells that remain, finding that a significant number of these resistant acral melanoma cells persist following long-term treatment of CDK4/6 inhibition. And then we also see that using EDU, which is a proliferative assay, we find that compared to 3 days of treatment where there's an initial decrease in that proliferative capacity of the population, if you continue treating those acral melanoma cells chronically beyond 45 days, you see that much of the population reactivates that ability to proliferate. And so obviously we wanted to understand how could this be. Before getting into the potential resistance mechanisms, now that we had the therapy sensitivity information from the MTT, we went back and wanted to see, is there any correlations between CDK4 protein expression and other markers that have been recently published to serve a role? So we tested the protein expression of CDK4, CDK6, and cyclin D based off of that original paper that justified the clinical trial. We checked this same group that published 2017. They later, during the clinical trial, admitted themselves that this had no correlation, and then they came up with a new predictive profile. So we wanted to see whether this increased MCM7 and decreased SH2B3 would be predictive in our model system. And then, of course, in the acral melanoma field, there's not much that's been published, but it was recently published that these proteins Acral and LZTR1 are potentially drivers of acral melanoma. So we wanted to see, are they playing a role potentially in our sensitivity? So first, we looked at the protein expression on the y-axis and the IC50 sensitivity on the x-axis, and we did not see correlations with CDK4, CDK6, or cyclin D protein expression and the sensitivity to CDK4/6 inhibition. We also then checked using RNA-seq whether baseline expression at the mRNA level of these different markers might correlate with sensitivity. We found that MCM7 did not show any significant correlation with sensitivity across 3 different CDK4/6 inhibitors. This is palbo, abema, and ribociclib. We also did not see any significant correlation between SH2B3 mRNA expression and sensitivity at the inhibitors. Here, checking now LZTR1, which was recently published to be involved in metastasis for acral melanomas. Again, we did not see a significant correlation, and Acral has been proposed to play a role potentially in initiation of acrals. And again, we did not see any significant correlation with sensitivity. So then what could possibly be driving this resistance escape? So to identify this, we performed some PDX studies of acral melanomas. So here's 3 different acral melanomas treated with vehicle in black or with palbociclib in red. As you can see, there was maybe some modest sensitivity, but overall these tumor cells continued to grow with single agent CDK4/6 inhibition. So we collected protein lysate from these in vivo treated tumors, performed reverse phase protein array. And so anything in red are proteins that were increased in the context of escape. And we see a lot of old friends from the cutaneous melanoma field. So various members of our team are looking at the roles autophagy might serve because of the increase of these canonical autophagy genes, ATG4, ATG5. Looking at the ability of the mechanotransduction pathway to drive resistance, the ability of AKT-mTOR signaling given the hyperactivation of phospho-AKT. So these are 3 PhD students in our laboratory. And then Kaz fleshed out the opportunity of MAP kinase pathway potentially in this escape mechanism. So Kaz performed a time course study of a panel of 4 different acromelanoma cell lines. She saw again, decrease of these cell cycle machinery. But when she looked at the MAP kinase pathway, there was evidence of a robust hyperactivation across our cell line panel, as well as downstream cyclin D expression. And the cyclin D was interesting to us because CDK4, CDK6, and cyclin D actually make a trimeric engine with cyclin D to promote cell cycle progression. So philosophically, it might make sense that the cells are increased in cyclin D when CDK4 and CDK6 remain inhibited. And so when looking in the literature, the hyperactivation of the MAP kinase pathway has been seen before in the context of acquired CDK4 inhibitor resistance. And so this has been seen in breast cancer and uveal melanoma. And in each of these cases, you add a MEK inhibitor, you increase the efficacy. What was not known previously is how within the 72-hour window, hyperactivation of the MAP kinase pathway might occur from CDK4/6 inhibition. And so this was an area of unknown. Our laboratory wanted to figure out what the mechanism might be. And so as we tried to figure out a plan of how to go about understanding this, we first were inspired by what was previously published more than a decade ago from the Neil Rosen laboratory. And in this context, they found that BRAF mutant superficial spreading melanomas have such hyperactivation of phospho-ERK that it leads to increased expression of these MAP kinase pathway negative regulators, DUSPs and Sprouty. And if you use a BRAF inhibitor, you actually block the expression of DUSP and Sprouty. The expression is reduced, and now the MAP kinase pathway is primed to be reactivated. So we wanted to see in our different system, a BRAF wild-type system with a CDK4/6 inhibitor, would there potentially be rewiring of these negative feedback regulators? And so Kaz, looking at her time course lysate, could confirm of all the different negative regulators she looked at that there was a robust decrease of DUSP4 protein expression across our cell line panel. To determine whether it was functional, Kaz overexpressed constitutively the DUSP4, and then determine whether or not adding palbociclib when DUSP4 was overexpressed would have a significant difference. And indeed, she saw that you could reinforce the inhibition of the cell cycle inhibition of palbo when DUSP4 was overexpressed. You block that phospho-ERK hyperactivation. And then functionally, when you look at the proliferative capacity of these cells using EDU, you get the most significant inhibition of proliferation when you overexpress DUSP in the context of palbo. We then wanted to see downstream of the MAP kinase pathway hyperactivation, what was functionally driving this escape from therapy. And so Kaz next explored the induction of cyclin D that she saw. And so cyclin D has been previously shown in other types of melanoma to be driven by the MAP kinase pathway, but it's never been explored or confirmed in Acral. So Kaz took these acral melanoma cell lines. She first serum starved in the absence of FBS. She added back FBS. You see that there's hyperactivation of ERK. There's induction of cyclin D. And then in this context of refeeding, she added a MEK inhibitor or an ERK inhibitor, showing that she could block that induction of cyclin D, just showing that at least in part, the MAP kinase pathway dictates cyclin D expression in acral melanoma. So next, she wanted to silence cyclin D to see whether or not it functionally played a role in palbociclib efficacy. And long story short, you get the most robust inhibition of proliferation when you silence cyclin D in the context of palbociclib treatment. And this also significantly increased the ability of inhibiting proliferation, as seen by the greatest decrease in EDU positivity. So we next wanted to understand, does this have potential translational utility? Could we add a clinical grade MEK inhibitor, trametinib, to that palbociclib to increase the efficacy against these acral melanomas? And so here we treated a panel of different acral melanoma cell lines and indeed saw the greatest inhibition of cell cycle machinery when you add trametinib to the CDK4/6 inhibitor. You get the greatest induction of apoptosis, as evidenced by cleavage apart. Kaz created these 3D spheroids of acral melanoma. You see a greater cytotoxicity from the combination versus the CDK4 alone. In general, it seems like much of the cytotoxicity occurs from the MEK inhibitor, while much of the cytostasis occurs from the CDK4/6 inhibitor. So that combination is what seems like it's a sweet spot in terms of anti-tumor activity. She saw that long-term durability was increased in these 3-week long chronically treated colony formation assays. And then also the greatest decrease in the proliferative capacity with the combination versus either single agent alone. So next, Kaz went in vivo to see whether this could also significantly increase the anti-tumor activity of palbociclib. So using the WM4223 model, which was established from the left foot of a patient, she saw that single agent of MEK inhibition or CDK4/6 inhibition each had effects, but the most durable, long-lasting effect was with the combination. Using a second model of Acral UC, which was isolated from a left heel of a patient, we also see the same phenomena of the greatest durability from the combination. And then just confirming by Ki-67, you get the greatest decrease of proliferative capacity in the tumor tissue when you combine the combination versus single agent alone. And so based off of this data, we're trying to work with Crystal Legoo, a dermatologist at Hopkins, to try to launch a potential clinical trial for these patients. We next looked into the context of acquired CDK4/6 inhibition resistance and to see whether or not the hyperactivation of the MAP kinase pathway may also be playing a role. And so for this, Kaz chronically treated a panel of 4 different acral melanoma cell lines for up to 2 months with increasing doses of palbociclib. She checked and verified that they were resistant to palbociclib in red versus the therapy-naive parentals in black, and they were also cross-resistant to ribociclib. And then when looking at the protein level, she saw a remarkable hyperactivation of phospho-ERK in each of the CDK4 inhibitor resistant cell lines compared to their therapy-naive parentals. So we wanted to try to understand what could be driving that hyperactivation of the MAP kinase pathway in the context of acquired resistance. And first, we looked for inspiration from the breast cancer research that has found a lot of different potential mechanisms for acquired resistance to CDK4/6. And so we checked whether there might be changes in ERBB2, which has been found to occur in ER-positive breast cancer. We did not see any evidence in our resistant versus parental pairs. We checked whether AKT mutations or FGFR2 amplification or NRAS mutations might occur because of evidence in HR-positive breast cancer. We could not corroborate those findings, at least in our acral melanoma system. But what we did see is a return back to those negative feedback regulators. And in this context, DUS4 was not what was robustly decreased, but here now Sprouty, which acts at the level of receptor tyrosine kinase inhibition. And so Kaz has begun trying to explore mechanistically whether Sprouty is what's really driving this hyperactivation. And so far, when she overexpressed Sprouty, at the very least, she sees that there's a decrease in the MAP kinase activity. And so ongoing studies are trying to see the consequences on proliferation. But again, testing the utility of adding a MEK inhibitor in this context of acquired resistance, when we add trametinib to these CDK4 inhibitor resistant cells, we can induce apoptosis. We can significantly prevent the growth in these 3-day MTT assays. We could induce cytotoxicity in these 3D spheroid approaches, increase the durability of CDK4/6 inhibition, and again, reduce that proliferative capacity by EDU staining. And then finally, we wanted to understand, just checking in vivo whether or not this would also have utility. And so here we took chronically treated cells, we injected them into the mice, gave them a brief holiday to allow tumors to form. And when they were palpable, we again challenged them with the MEK inhibitor, the CDK4 inhibitor, or the combination. And long story short, we found that compared to what each of the agents might accomplish by themselves, the most durable effect was in the combination. You might see that the CDK4/6 inhibitor had some effect again. And at first, this was puzzling to us. But, given the literature, there's been evidence that CDK4 inhibitor resistance could be either permanent or reversible, depending on if it was genetic or non-genetic. And so given we gave this tumor cell line a holiday, we believe that at least in this context, it's a reversible resistance mechanism. And then in a final series of experiments, we wanted to try to understand what's the mechanism of action of that combination in vivo. So we performed reverse phase protein array. And what we found was that the combination of a MEK inhibitor plus a CDK4/6 inhibitor gave the greatest signature associated with DNA damage, the greatest decrease in CAMP, the greatest decrease in PARP, as well as RPA32. There was a signature associated with the greatest induction of cell cycle arrest, and a signature associated with the greatest induction of apoptosis, as evidenced by increases of these BCL2 proteins, as well as the anti-apoptotic BCL2A1, a significant decrease. All to say, in summary, what we have, I hope I have convinced you, is that patients with acral melanoma suffer poor outcomes, and the metastasis and therapy resistance landscapes remain poorly described. We hypothesize that the plantar skin microenvironment is promoting metastasis and therapy resistance. We find in the clinical data and our preclinical work that these CDK4 pathway alterations common in acral melanoma, but the CDK4/6 inhibitor does not confer durable clinical efficacy. We find in our system that the hyperactivation of ERK following acute and chronic treatment to these CDK4/6 inhibitors may be a resistance mechanism that can be an actionable opportunity to increase the efficacy. And use of a MEK inhibitor increases that cytostatic and cytotoxic effect of the CDK4/6 inhibitors. And with that, some future directions that we have are trying to understand the biological drivers of racial disparities. understanding the role age plantar skin might be serving in acral melanoma biology. I didn't show it today, but that same orthotopic model for metastasis, we had those were young mice. We've done the same experiment with age mice, and interestingly see that the metastatic site changes depending on the age of that environment. And we're also trying to define the epigenetic mechanisms that underlie that therapy resistance and metastasis. And with that, I want to thank my laboratory, a team full of great people that are doing excellent science, our collaborators. Casey's been helping out a great deal, giving us models. We really appreciate our funding. And with that, I will take questions. I think I'm supposed to have that slide. Thank you.
Couple questions. The second one, you kind of you alluded to at the end, but just going to ask the aging question.
Yeah.
But the first one is, so these are initiating this in the plantar skin. Is it a different stem cell or progenitor cell population than the ones that would be where you normally would get the cutaneous melanoma?
There's been some work characterizing the melanocytes at those different anatomic sites. There's evidence that the melanocytes in the foot adopt a very distinct transcriptional program, and they have overexpression of a receptor that is known to bind to certain nerve growth factors. How that serves a role in the differences in metastatic capacity and aggressiveness, that's to be explored. But because of those differences at the transcriptional level between melanocytes of the foot versus other sites, people feel those are essentially two different cell types, making acral melanoma essentially a different disease. It shares a name, but it's truly a different beast. So it's a great question and something that needs to be figured out.
The other one related to aging, you know how that collagen matrix changes with age? I'm sure you think about this with Ashley.
Yep. Yeah, we have a PO1 that unfortunately get funded, but looking at, in the context of cutaneous sites, how the collagen matrix and the stiffness of the tissue changes. And so it's not something that's been well published. Ashi's published showing that collagen abundance decreases as you age, but the abundance of collagen doesn't necessarily predict the stiffness of the tissue. We have some preliminary data showing that the stiffness may be increasing as you also age, but very interesting questions that need to be explored further. Thank you. No, those are great questions. I mean, these are all wonderful questions. Yeah, very important questions. In terms of potential microbiota differences, I mean, all the work that Casey and Richard are doing in terms of mucosal and the microbiome are very exciting. And hopefully at some point, we'd like to see how the microbiome, which is likely different in the skin of the foot versus other sites. In terms of the general microenvironment, we have studies, that I showed looking at first the biomechanical differences of that microenvironment, so the stiffness, the collagen density, the orientation of the hair follicles, the absence of them, and that glabrous skin of the foot is an excellent point that is worth investigating. We're also looking at how cell types in the plantar skin microenvironment may have different functions or potentially be more pro-tumorigenic compared to those same cell types in non-plantar skin. So those are some of the ways we're approaching it. The hair bulb we haven't thought of yet, but that's a great idea to try to understand differences for that stem-like pool and how that might be contributing, but lots of work to do.
So the models that you did during your objective, the blood versus the blood, saw huge differences in metastasis. And I'm wondering, given the fact that in humans you have metastasis primarily to the lymph nodes you were mentioning, wondering if you see any differences in lymph vessel sort of into the tumors in the foot versus the blood.
That's a great question. We have now somebody in the lab that's able to at least isolate those tumor draining lymph nodes. So that's something we're gonna look into. We haven't checked yet, but like you said, I mean, in the human evidence, it seems like that's one of the main sites is that in transit lymph node metastasis. So that's something we're looking into to see the difference. A great question.
Kind of what this works on, but are there other risk factors, non-age and ethnicity, like lifestyle, alcohol, anything else we correlate with?
That's a great question. There was some thought that potentially injury and trauma could serve a role. My understanding is there was a study in a village where footwear wasn't always worn between people. But perhaps maybe because the number was so small, significant differences between footwear and not footwear were unknown. Whether there's differences in how active you are and running, I mean, there's likely something out there. It's just the numbers have been so small at the moment that any concrete correlations, to my understanding, are not known. But all excellent things. And there was one study starting to look into a model where they had mice walking on a wheel. And they saw, which you might predict, walking would activate the YAP activity and mechano signaling. So our laboratory at the moment have ordered wheels. And as part of our thing is we're going to see whether these tumor-bearing mice, whether activity might also be dictating metastatic spread and resistance. So all interesting areas where we've never done it, but we're looking into trying to figure out the best way to see how activity might also change the biology of these tumors. Yes. So most people haven't heard of Acral, but everyone almost has heard of Bob Marley. And yeah, he had a tumor on the toe. They thought it was an injury from soccer, ended up being a metastatic acral melanoma. I think he was 34 years old. And yeah, that's what took him.
I guess I'm allowed to ask questions. In terms of the profile and what you see when you put the cells in the subcutaneous versus the foot, and you get this very different profile with the suction, what do you see like in the cell line? So when you culture it in vitro, is it more like the subcutaneous or does it look like? And then like once these metastasize to the liver, do they maintain that profile that from because they originated in the foot? Is it like it's done program? I'm trying to get at. I think this is so important when you think about testing these targeted therapies and testing treatments and what states these cells are in. So how does the state correlate, I guess, with the future and then metastasis?
No, that's a great question. And so far, when we're doing in vitro cell line stuff, it hasn't been obvious differences between therapy sensitivity or invasive capacity of these acral versus cutaneous. Something we're looking into is whether it's a nature versus nurture. Are the acral melanomas by their own intrinsic cell nature more aggressive than the cutaneous melanomas, or is there something in the environment of that foot that's nurturing them to a more aggressive state? And experiments that we have ongoing are injecting cutaneous melanomas into the foot as well versus the flank to see whether they will also show an elevated metastatic capacity. And it might just be a complete microenvironment driven thing. And in terms of whether or not they retain when they go to the liver, that's something we're exploring because there's this concept of mechanical memory that if you culture, at least in the stem cell field, stem cells enough on stiffness plates versus soft, after culture of 10 days or so, if you put those stem cells into a soft matrix, they retain the activity they had as when on stiff. And so we're exploring that mechanical angle in terms of the epigenetic reprogramming and how much of that is retained when they leave the primary site. But great questions. We're a lot of work to do. You got it, yeah.
DNA. So, the mutation profile is different compared to the cutaneous melanoma, but still they have some driver mutation. Right? So, do they have UV signature or the mutation type is different?
The mutations are considered not to have a UV mutation. There's a lot of structural rearrangement. There's a phenomenon that Boris Bashan's team has found called a typhona, that is 40% or so of acral melanomas have these massive structural rearrangements. Yeah. There's a little bit of, yeah. Yeah. So I think there's a little bit of debate of whether or not those 15 to 20% of BRAF mutant acrals, some I've heard believe they may be more similar to cutaneous than acral. We've tried to begun doing some PCA plot of acral melanomas with BRAF mutation versus cutaneous melanomas with BRAF mutation. We don't see a complete overlap. So how different or similar the same mutation across these subtypes drives the biology is I think the debate is still there to be solved. But yeah. Yeah. That's a great question. That's a great question. I think Casey also published not too long ago, probably looking very closely at the UV mutation spread across different subtypes of acral. Because I know there's been, from her work, showing pretty remarkable differences between the fingernail acral melanomas versus the foot versus the palm. So definitely a good question. With that, thank you. Yeah, thank you. Oh, there was some on the chat. Okay. Gotta be healthy, yeah. I know, it's hard to battle those two things.